Okay, so we're going to talk about our immune system, and the first thing we have to talk about for that is just to describe what a pathogen is, what an infectious agent is. You'll take a whole class on this if you're going into nursing or dental hygiene, so you take a whole microbiology class. So pathogenic means it causes disease. I need to splurge and buy myself a nice stylus, because I'm using these cheap ones and they don't seem to be super wonderful. Okay, so cause disease. And they have, sorry, I'm talking to myself about styluses. Okay, so basically there are 5 classes. So there are bacteria, which like staph, strep. There are fungi, and there aren't many human pathogens that are fungi. There are some, so something like a yeast infection. Protozoa, so those are small animals, so something like Giardia, an amoeba. Okay, multicellular parasites, so that would be like worms, something like that. And then viruses, and viruses aren't considered living. So an infectious agent, something's going to cause, a pathogenic infectious agent, something that's going to cause disease. Okay, now we are going to respond to them with white blood cells. So remember white blood cells. So remember leukocytes. Remember they're broken down into two kinds, agranulocytes and granulocytes. So you're familiar with those. Under the granulocytes, remember we have Ben. So we have basophils, eosinophils, and neutrophils. Okay, so here's a brief description of how they work, and we're going to go into a little more detail as we keep going. Okay, so neutrophils are kind of like the Marines. So they're the first on the ground, they're the first to arrive, and they can go in and they can kill cells. Okay, eosinophils maybe are a little more specialized, and what they can do is they can go in and they can take on parasites. So they can take on the big guys. Sometimes they also go up when someone is having an allergic reaction to something. Okay, now basophils are called basophils when they're in the blood, but if they leave the blood and go into the tissue, they're called mast cells. Okay, now mast cells engage in we call chemical warfare. So they're not going to engulf or destroy pathogens by themselves. They're going to release chemicals to help the cause. Okay, so I always think of it as chemical warfare. Okay, now agranulocytes are made up of monocytes and lymphocytes. So monocytes, when they leave the blood and enter the tissue, that's when we call them macrophages. And macrophages are big eaters, so they're going to go in and they're going to phagocytize something. They're going to gobble it up. Okay, now lymphocytes. Okay, so we have a bunch of different kinds of lymphocytes. We have T lymphocytes, which T lymphocytes and B lymphocytes are more like special forces. They're going to be very specific in what they kill. T lymphocytes kill specific kinds of cells, so they're going to look for like strep, and they're going to kill strep. B lymphocytes, these actually make antibodies. Okay, and the antibodies, we're going to kind of refer to the antibodies as a bomb. And they can go in, and the bombs are very, very specific. So the antibodies are specific against a specific invader. And then we also have these cells that are called natural killers. So I wrote killer. They can kill anything. So natural killers are not specific. They're just kind of crazy, and they're out there and they can kill any kind of cell. It always makes me think of Natural Born Killers, which is an old movie from like the 90s, which they're crazy. They just go out and kill whatever. Okay, now, where are the leukocytes found? So they're in lymphatic tissue. Okay, they're so in the lymph nodes, the spleen, tonsils. They're in organs in the body. Okay, so we have them in our lungs, in our brain, in our intestines. Okay, they're found in epithelial layers, so places where there are openings, so mucous membranes. So we call them dendritic cells when they're in mucous membranes. And then they're also in connective tissue, so those are mast cells. Okay, now cytokines. Cytokines, well, here's the definition. So they're small soluble proteins produced by immune cells. And what they're going to do is they're going to communicate. It's how cells communicate with each other. Okay, so if this is a cell and this is a cell, the way that they're going to talk is they're going to release these cytokines. And the cytokines are going to be released. Okay, they'll bind to a receptor, and then they're going to cause a change to occur in the next cell. So cytokines are going to regulate the behavior of immune cells. They will regulate inflammation. They can also serve as weapons. Okay, so there's like a couple categories of cytokines, and they know a ton about this. You can take a whole like graduate program on immunology. So you guys are just really dipping your toe into the immunology pool. Okay, now there are interleukins, which they refer to as ILs. Okay, so interleukins. So there's interleukin 1, interleukin 2. There are tumor necrosis factors. Okay, so they're going to, guess what they do? They kill tumors. Colony stimulating factors, which are going to stimulate growth. And then interferons. And interferons are released when you're exposed to viruses, sometimes cancer. Okay, now, immunity is the state of being protected from harm. Okay, so if you've been granted immunity from something, you're not going to be harmed by it. Okay, now, it's divided into two broad categories. Okay, and these are important. One of them is innate immunity, which is non-specific. Okay, it's against anything. So you're born with it. It protects you against a wide array of things, and it responds right away. Okay, so non-specific is also known as innate. Okay, so innate is non-specific. You're born with it. We'll talk about things like your skin. Okay, adaptive immunity is specific. Okay, so it's developed in response to different antigens. Okay, so it's specific to the antigen that you're being exposed to. Things that are involved in adaptive immunity are B cells and T cells. Okay, T cells take care of cell-mediated immunity. So the T cells are going to actually do the work to get rid of the pathogen. B cells release antibodies into the fluids of the body. And fluids are often referred to as humors. Okay, so fluids are referred to as humors. So the antibodies are released into the fluids or the humors, and that causes humoral immunity. Okay, this creates an immune response. It is not immediate. It can take several days. And it depends on how many times you've been exposed to it as to how long it takes. Okay, so here's a chart, or I don't know what, I guess a chart that kind of explains it. Okay, so here's the immune system. We have innate, which we're born with, right? And then we have adaptive, which is specific. Okay, so innate is an immediate response, okay? And then there's adaptive, which is delayed, but it's specific. Okay, so innate is things like the skin, the mucous membranes. They're going to prevent things from getting in. Okay, that's straightforward. That's easy. Okay, then we have non-specific internal defenses. So these are things that are just going to kill things that are non-self. Okay, things that look foreign. So we have macrophages, natural killer cells. We have chemicals such as interferon and complement proteins. And then we have physiological responses like inflammation and fever. Okay, so we'll talk about these in this set of notes. Then the next set, we're going to talk about these. Okay, so adaptive immunity is T cells and B cells. B cells, I want you to remember they make antibodies. Okay, so here's preventing entry. You guys can look over this slide and read it. I think it makes a lot of sense, right? You can look at all the barriers that we have in our body and how they protect us from pathogens getting in. That is part of your innate immune system. Okay, so it's all innate. You don't have to think about it. You're born with it. It's right there. Okay, now, if something does get in, how are we going to fight against it? Okay, so neutrophils we're going to talk about first. Okay, so neutrophils, these are the most prevalent white blood cell in the blood. They are the first to arrive if you have inflammation. Okay, them and macrophages kind of work similarly. Okay, they work in the same way. Okay, so macrophages, they arrive after the neutrophils and they stay longer to kind of clean up the mess. Okay, so how do they protect? The way that they protect is they phagocytize bacteria. Okay, so they come in, they engulf it. Okay, they're phagocytic cells. They come in, so here's the infectious agent. They engulf it. It merges with a lysosome or a phagosome. Okay, the phagosome merges with the lysosome. The lysing enzymes destroy the pathogen, and then we get rid of the waste. Okay, same kind of thing happens with dendritic cells. So they phagocytize bacteria. Sometimes they create a killing zone around them where they actually release some of those phagocytic or some of those chemicals that'll break things down, and then that'll destroy the pathogen. Okay, so phagocytic. Okay, now basophils and mast cells are different. Okay, they're different. They have, these are the ones that we say they're like chemical warfare. Okay, so what they do is they're going to secrete histamine and heparin. And I think we've talked about those in blood when we talked about blood. So histamine makes capillaries leaky. Okay, so it makes capillaries. Okay, so we talked about histamine and heparin before. So histamine makes the capillaries leaky, and heparin is it keeps blood from clotting. Okay, so heparin's going to keep the blood from clotting. So if we look here, here's kind of what basophils and mast cells do. So here's the basophil. It's releasing histamine. Okay, it's releasing heparin. So it's making the blood more permeable. Okay, or the capillaries more permeable, so fluid can leave. And then the heparin is serving as an anticoagulant to keep the blood flowing around the site of the wound. Okay, the eicosanoids are a type of cytokine, and they will increase inflammation. Okay, the big thing that I want you guys to get is I want you to understand that basophils are, basophils secrete chemicals. They are not going to be phagocytic. Okay, now, natural killer cells. So natural killer cells, they're going to circulate in the blood. Okay, they're formed in the bone marrow. They circulate in the blood. They accumulate in the lymph nodes, the tonsils, the spleen. They do immunological surveillance. So basically, they're hanging out looking for things that might be infected, so like virus-infected cells, bacteria-infected cells. They're looking for tumor cells. They're looking for transplanted tissue. So they're looking for things that are not self, so things that don't look like they belong. Okay, now the way that they kill is pretty slick. Okay, so what they do is they actually insert this protein called perforin. And perforin, if you think about perforated paper, so perforated paper. Okay, so if you think about that, it's going to put holes, okay, into the unwanted cell. Okay, and then it also inserts an enzyme called granzyme, okay, and this causes the cell to break down and for the cell to die and go through apoptosis, which is programmed cell death or cell death. Okay, now eosinophils, they're protecting against the big things. So here's a parasitic worm. Okay, and what they do is they have antiparasitic effects. So they'll accumulate around the parasite and release chemicals or enzymes that are going to help digest the worm. Okay, eosinophils, they're going to break down or phagocytize antigen antibody complexes. They can phagocytize allergens, and they can also attack parasites. Okay, now interferons, these are chemicals that protect. Okay, so these are proteins that are going to be released. So interferons are antiviral. So what they're going to do is, and this picture makes it, I want it to be simple for you, and this picture from the book looks more complex. But what they do is the interferons are released from a cell that's infected. So they're released from a cell that's infected with a virus. So here's the virus on the surface of it. Okay, and what they're going to do is they'll tell, so these little red dots are supposed to be the interferon. They can, one, activate natural killer cells. Okay, and the natural killer cells go around and they'll look for cells that are infected with the virus and they'll kill them. Okay, they can stimulate the production of macrophages to go around and engulf the cells. And they can also tell the other cells what's going on so that they make more antiviral proteins so that they can't get infected. Okay, so interferons produced by virus-infected cells, and then they're going to communicate the fact that, yes, we're infected by a virus, and this is what we need to do. Okay, now the complement system. So the complement system is a series of proteins or a group of proteins. They're made by the liver. Okay, so that's important. But they're activated in the blood. So they're made by the liver and they're released into the blood. Okay, there's two pathways for the complement system to work. One is called the classical pathway, and that gets started when a complement protein binds to an antibody, and that antibody's attached to an antigen. Okay, now the alternate pathway is when polysaccharides, okay, so carbohydrates from a pathogen bind with the complement protein. Okay, and that turns it on. Okay, now what does the complement proteins, what do they do? So there's 4 things that they do. And you can memorize it by going ICE. Okay, so first thing is opsonization. So the protein binds to bacteria, and that's actually going to enhance phagocytosis. Okay, so it makes the bacteria, okay, so if this is the bacterium, some of the complement protein can bind to it, and it makes it easier for the organisms to recognize and break down. Okay, so it kind of like it enhances phagocytosis. Okay, now it can also cause inflammation. Okay, so inflammation is going to be enhanced because mast cells and basophils are activated. So they're going to start releasing more histamine and heparin. Okay, they can cause cytolysis. So they insert this thing that's called a membrane attack complex, and that basically puts holes into the to the membrane of the organism or the pathogen. Okay, and then it's also going to help us get rid of immune complexes. So when an antibody binds to a pathogen, it'll help us get rid of it. Okay, so here's pictures of what was just written out. Okay, so opsonization, the complement protein are the blue things that are bound to here. So they bind to it. It makes it, it almost makes it like it tastes better, and it's easier for the macrophages to phagocytize it. Okay, the complement proteins also stimulate mast cells, basophils, neutrophils, and macrophages. Okay, so that's going to start kind of enhance inflammation. Okay, breaks down cells. Okay, and then it helps eliminate these immune complexes. Okay, so I just want you to know the basics. They know way more than this, but we just want to know the basics. Okay, now fever. So fever is part of your innate immune system. Okay, so words that deal with fever are like pyrexia, so like pyromaniac, right? Fire. Okay, and then febrile. So things like Tylenol are considered antipyretics. So a pyretic causes a fever, antipyretic would stop it. Okay, so fevers are a defense mechanism, so they're going to help by when your body temperature goes up, it's harder for the organisms to survive. Okay, so the way that a fever works, okay, is You get infected, okay, and the macrophages engulf the bacteria or the virus, and then they secrete a pyrogen, which is interleukin-1, okay. It resets the thermostat in the hypothalamus, okay, and your body has to warm up, okay. So basically then you get hot and cold and hot and cold, and you try to raise your body temperature. So that's onset. Okay, stadium is when the body temperature fluctuates or oscillates around the new set point. Okay, and then when the fever breaks, the infection ends, and then you probably are sweaty and your body cools down, and that's called defervescence. So you have infection, pyrogen secretion, hypothalamus, thermostat gets turned up, onset, stadium, infection ends, okay, and then defervescence is when we go back to normal. Okay, so a little bit of a fever is not a bad thing. It helps fight infection, but if you get greater than 105, that can cause you to be delirious. 111 to 115 can, a positive feedback loop can kick in, and that'll lead to coma and death, and proteins will denature and you can die.